thank you so much for the invitation and thank you for the introduction. And so today I will present you the work that I have done in Stefan Pless lab at the University of Copenhagen. And during this work, we have used a protein semisynthesis, and I will explain you which type of technology we have used later in order to study NaV1.5 channel. And the aim was to study more the intracellular conformational landscape and regulations of this ion channel. And an outline of the presentations of today. So I will start with a really brief introductions on NAV 1.5 and also on the technology that we use. And then I would like to present two different projects that we have performed with this type of technology. And in the first project, we have studied the phosphorylation sites on NAV 1.5 intracellular domain and how it impacts the intracellular protein-protein interaction. And on the second project, with the same methodology, we have incorporated a fluorescent dyes in order to follow in real-time molecular motions that happen during inactivations in living cells, so still for NAV 1.5 intracellular domain. So NAV 1.5, they belong to voltage-gated ion channel superfamily that is represented here. And this superfamily contains several members with distinct structure, distributions, and physiological role. And if we talk about the voltage-gated sodium channels, there is 9 different members that differs in their tissue distributions and physiological function. And among them, NAV 1.5 is distributed mainly in cardiomyocytes, where it triggers the initial phase of the cardiac action potential by driving this fast sodium entry happening after depolarization. And NAV 1.5 dysfunction is associated with a lot of different cardiac diseases. And just to name a few, there is, for example, the long QT syndrome. There is also the Brugada syndrome, the sick sinus syndrome, and also several types of arrhythmias. And in clinic, NAV 1.5 can be modulated by antiarrhythmic drugs. So there, I think it demonstrated that NAV 1.5 is a really important therapeutic target to study. In terms of structure, NAV 1.5 is a pseudotetramer of more than 2,000 residue, and this constitutes what is known as the alpha subunit that is represented here. There is also 4 domains that are connected by intracellular linker, and the N and C termini, they are intracellular. And here you can see the C terminal domain of NAV 1.5. And importantly, between the D3 and the D4 domain, there is this really important part of the channel that is the D3-D4 linker. And this D3-D4 linker, it contains this tripeptide here, which is known as the IFM particle and sometimes also named the inactivation particle. And this part is really crucial for NAV 1.5 activity. In terms of activity, the function of NAV 1.5, it depends on conformational changes that are triggered by variations of the membrane potential. So at a resting membrane potential, NAV 1.5 is in a closed state. And after depolarizations, there is a fast conformational change that occurs, which provokes the opening of the ionic pore and the passage of sodium. And there, really fast after opening, there is another conformational change that occurs in a millisecond range that is known as fast inactivation. And this terminates the sodium flux. And this is done by allosterically triggering the closure of the ion pore. So on this figure, the IFM is represented by occluding the pore, but this is not the case. It's not directly occluding the pore. It's an allosteric closure of the pore. Inactivation can also occur from a closed state via a transition that is known as steady state inactivation. And the precise timing and the precise voltage dependence of inactivations, they are really essential for maintaining a normal cardiac excitability. Differences of only few milliseconds or only a few millivolts in the voltage dependence can have a dramatic effect and cause severe arrhythmias and also cardiac diseases. And in terms of structure, the intracellular domain, they are really important for inactivation, and notably the D3-D4 linker, but also the C-terminal domain. NAV 1.5 is gated by voltage, but its activity is also highly regulated and modulated by other factors. And among them, there is the post-translational modification. So those can be glycosylation, sumoylations, methylations, but also phosphorylations. And we are really interested by phosphorylation because there is more than 40 phosphosites that has been identified in NAV 1.5 intracellular domain. And an interesting point is that it seems that the phosphorylation level of NaV1.5 seems to be elevated in heart failure and oxidative stress, so in pathological condition. But there is also some associated or auxiliary proteins that can affect NaV1.5 activity. They can form a macromolecular complex with the alpha subunits, and they can modulate the localization, the expression, and the function of NaV1.5. And interestingly, the variant of these auxiliary proteins, they are also associated with cardiac diseases, which highlight their really important role. So to summarize a bit, the intracellular domains, they are involved in activation, and they can also be affected by phosphorylations and also the binding site for several cytosolic proteins. But it's quite difficult to study intracellular domain of ion channels. They can be difficult to target, and that's why we have decided to implement a semi-synthetic method to be able to study the intracellular domain of NaV1.5. And the semi-synthetic method that we use is known as tandem protein transplicing, and I will explain you what it is now. So this is a semi-synthetic approach, and it relies on the use of two orthogonal split intein pairs. So these are represented here in purple and here in pink. And these split intein, these are naturally occurring protein sequences that are able to undergo a spontaneous splicing reactions when they colocalized. So to apply this technology to NaV1.5, this tandem protein transplicing, we have divided the channel into three parts. There is first the N part that contains the D1 to D3 domain, fused with one part of the split intein, the first split intein. There is the C part that contains the D4 and the C terminal domain fused with the second part of the second split inteins. And there is the X part that correspond to the D3-D4 linker that is fused with the other part of the split inteins. And those parts of the channel can be injected into Xenopus levis oocytes. The N and C part can be injected as RNA, and the X part can be injected as a peptide. And this allows the splicing reaction to happen and the expressions of the full-length channel at the membrane of the oocytes. And this formation can be followed by electrophysiology. And there is only the injections of the three components of the reaction that can result in NaV1.5 full-length expressions. The injections of only N and C parts does not lead to the formation of the channel, which is exemplified here because there is no current that is evoked by a change of potential. And the really crucial point of this technology is that this peptide X is synthetically accessible. So it means that you can introduce any type of probes or modifications that you want in the intracellular domain of NaV1.5 and more broadly on any protein of interest. And with that, in the context of NaV1.5, we decided to introduce first non-hydrolyzable phosphomimic, which will correspond to the first project that I will present you. And second, fluorescent dyes. And more precisely, we have incorporated them into this alpha helix that is just upstream of the IFM particle of NaV1.5. And now I will present you the first project. And in this project, we have introduced a non-hydrolyzable phosphomimic into NaV1.5 D3-D4 linker. So the idea there was to study tyrosine phosphorylations because it has been proved that it regulates NaV1.5 activity. And more precisely, to study this site, this Y1495 tyrosine phosphorylation site in the D3-D4 linker because it has been seen in vitro that it was important for inactivation. But phosphorylations, it's quite difficult to study directly because in the cellular context, tyrosine can be phosphorylated by kinases and they can be also dephosphorylated by phosphatase. So it leads to a dynamic equilibrium of proteins with phosphorylated and non-phosphorylated proteins with no control on the amount of phosphorylated protein that we observe. So in order to study the impact of these phosphosides in a non-ambiguous manner and in a precise manner, we used a non-hydrolyzable phosphorylated derivative. And this is known as a phosphonylated tyrosine. And you see that this oxygen bond here is replaced with a carbon bond, which means that this component here is non-hydrolyzable by phosphatase. So this allowed to express a populations of proteins that are 100% phosphorylated at the site of interest. And to use as a control, it is possible to use a phenylalanine instead of tyrosine because it cannot be phosphorylated. So to do tandem protein transplicing, the peptide X, they have been synthesized with these derivatives. And this project has been started by Jacopo Galeano, who was a postdoc in the lab, and Hendrik Arms, who was a PhD student and then a postdoc in the lab. And they have followed by electrophysiology the steady state inactivation voltage dependence on the non-phosphorylated and the phosphorylated construct that they have been able to reconstitute via tandem protein transplicing. For the non-phosphorylated construct on Y4095, they have seen that it has a wild-type-like inactivation voltage dependence. For the phosphorylated constructs, it reveals that it shifts the steady-state inactivation properties to more depolarized potential, and it elicits late current. So they have been able to put into evidences directly that these phosphorylation sites have a critical effect on NaV1.5 function, and namely on inactivation. Then our collaborators at the University of Canberra in the group of Ben Corey, they have performed some molecular dynamic simulations on the full-length NaV 1.5 model that they have obtained by computational approaches. And there they have been able to see that Y49 phosphorylation, represented here in purple, it was increasing the relative distances that can adopt the C-terminal domain versus the D3-D4 linker. Because you can see here that there is a second conformation that can be adopted when this NaV 1.5 is phosphorylated, and this is displaced by approximately 10 angstrom. It shows also, because there is several conformations that can be adopted, that there is an higher conformational plasticity of this intracellular domain with the phosphorylations. So it highlights really the critical effect of these phosphorylation sites on NaV 1.5 function and activity. And because this phosphorylation was affecting the C-terminal domain D3-D4 linker distance, we were interested to look at CTD interacting proteins. And we decided to look further at this family of protein, this FGF family, which is a protein or family of proteins that interact with the globular domain of NaV 1.5 C-terminal domain. And the member of these families, they have been described to have several effects on almost all the subtypes of voltage-gated sodium channels. And these effects, they include increasing the peak maximal current, shifting the voltage dependence of steady-state inactivations to more depolarized potential, and also decreasing the late current on variants that elicit late current. And we were interested to test if these interactions with the FGF family or these interactions between the FGF and NaV 1.5 was regulated by our phosphorylation site in the D3-D4 linker. That's why we decided to perform a systematic study of the main FGF isoform on NaV 1.5. So just to say a few words on this family, these are known as fibroblast growth factor homologous factors, and they can be called FHF or FGF or IFGF. So you can find all these names in the literature. And we have decided to look at A-splice variants, which contain the calmodulin binding motif, and also at B-type variants, which did not contain it. To talk a bit about their distributions, FGF12 and FGF13, they are expressed in human cardiomyocytes, and FGF14 is more expressed in the brain. And as I have said, they are binding NaV 1.5 C-terminal domain and modulate the function by depolarizing the shift, by provoking a depolarizing shift on steady-state inactivations and reducing late current. So to start the project, we have co-expressed wild-type NaV 1.5 with FGF12A and follow the steady-state inactivation properties. And when we compare the wild type with the wild type co-expressed with FGF12A, you can see that there is a shift in the steady-state inactivations as what was expected from the literature. And we have performed the same experiments with the other isoform of FGF. And these experiments reveal that they induce an isoform specific. You can see a different extent of shift on the depolarizing shift in the voltage dependence of steady-state inactivation. But then we were interested to look if this functional effect was impacted when Y4095 was phosphorylated. So to study that, we have co-expressed again the non-phosphorylated and the phosphorylated construct with and without FGF12A. And on the non-phosphorylated construct, there is a shift in the steady state inactivation that is observed, which recapitulates what we see on wild type. But with Y1495 phosphorylated, there is no further shift that is evoked. You can see here that the curve with and without, they are superimposed. So it means that this shift only occurs in absence of these phosphorylations at this site. This effect is also seen with FGF13A and FGF14A, where there is no further shift, as you can see here and here, when Y1495 is phosphorylated. The results are less clear for the B type, where you can see that the shifts they are a bit smaller, so we don't really know if it's in the error bar or if it's if the shift is also abolished. So this experiment demonstrates that this steady state inactivation shift only occurs in absence of the phosphorylations at Y1495 with A-type FGF. So it highlights the regulations of a protein-protein interaction by these intracellular phosphorylations. And just to say it a bit in another word, it seems that this phosphorylation is able to regulate the effect of FGF on NaV1.5 in a switch-like manner. On the non-phosphorylated NaV1.5, the switch is on, it has an effect. And on the phosphorylated NaV1.5, the switch is off and it doesn't have an effect on steady state inactivation. But then we wanted to assess if this phosphorylation was modulating also the effect of FGF on the late currents. And we have done that by measuring the late current on wild type, co-expressed with FGF12A, on a non-phosphorylated construct, co-expressed with FGF12A. And here you can see that there is no real differences in the level of the late current that is calculated. Then the phosphorylated construct is eliciting a large late current. And this is drastically reduced with the co-expressions of FGF12A. And this is also the case with FGF13A and FGF14A. Concerning the B subtype, this is also the case with the 12B subtype, but in a lower extent. But FGF13B and FGF14B, they are not able to reduce the late current provoked by Y1495 phosphorylation. And this experiment, it revealed two other interesting findings. So first, because we see this effect on the late current, even if before we have not seen any effect on the steady state inactivation properties, it means that FGF, they remain active on the phosphorylated construct. And secondly, it means that this shift in steady state inactivations and the late current modulations, they are likely to involve two distinct mechanisms. So to summarize what I have just said, Y4095, it impairs the FGF-induced shift in steady state inactivations, but not the late current reduction. We have seen in the previous studies that it destabilized the binding of the IFM particle in its receptor site, and that's probably what it's leading to late current. And also in the molecular dynamic simulations that it increases the distance between the CTD and the D3D4 linker. And to go a bit further on that, we wanted thus to destabilize the CTD-D3D4 linker interaction. And to do that, we have used the pathogenic variant, this E7084K variant, that is located on the C-terminal domain and acts by destabilizing the CTD-D3D4 interactions. So we have co-expressed again the non-phosphorylated and the phosphorylated construct that have these variants, and also with FGF12A and FGF13A. And when we look at the steady state inactivation properties with FGF12A, this time we have not been able to see any shift either on the phosphorylated but also on the non-phosphorylated construct. And we have observed the same effect with FGF13A. There is no shift on the non-phosphorylated and no shift on the phosphorylated construct. When we looked at the late currents, it is still reduced by FGF12A and also by FGF13A. So this experiment really highlights further that there is two distinct mechanisms that are able to perform the FGF regulations on NaV1.5. And then finally, we wanted to interrogate the importance of the domain of FGF. We know from the literature that is the core of these proteins that are interacting with the C-terminal domain of voltage-gated sodium channel. And we have seen in our experiments that the B-type, they seem less effective at reducing late current than A-type. So we were wondering what is the importance of this N-terminal domain that is different between the A and B-type. And in order to study that, we have co-expressed the intein phosphorylated and non-phosphorylated construct with a peptide that corresponds to the N-terminal domain of FGF13A. And this reveals again a shift in the steady-state inactivation properties on the non-phosphorylated construct and no further shift on the phosphorylated construct. The shift extent on the non-phosphorylated construct is smaller than when we co-express with the full-length FGF13A. So it seems that the N-terminal domain cannot shift as much as the full-length FGF13A. Then we looked at the late current, and this time the N-terminal domain of FGF13A was not able to reduce the late current. So it seems that full-length FGF is necessary to reduce this late current. So to conclude on this project, we have been able to put into evidences that this family of proteins, FGF, they regulate NaV1.5 via two distinct mechanisms. One that is this effect that we see on the shifting the voltage dependence of steady-state inactivations, and this one is dependent on the phosphorylation state. And the second, the reductions of the late currents, is independent on the phosphorylation state. We have seen that the destabilizations of the CTDD3D4 linker interactions, it abolished the FGF effect on steady-state inactivations, but not on late current, and that full-length FGF is required to reduce the late current. So we could say that this phosphorylation site is able to tune the effect of FGF on NaV1.5. And just to put it a bit in a more physiological context, these FGF expressions and also phosphorylations, they are both factors that are upregulated in pathological conditions. For example, in heart failure, some FGF, they can be upregulated and phosphorylation also. So it could be a physiological way of calibrating NaV1.5 activity in these different conditions. But then I also want to say that with this project, we have studied only one phosphosite over more than 40 phosphosites that has been discovered so far on the intracellular domain of NaV1.5. And also that there is numerous other post-translational modifications that can happen. So it is just highlighting further the huge complexity of NaV1.5 regulation. And with that, I would like to continue with the second project. And in this project, this time we have introduced a fluorescent label, so this rhodamine dye, into NaV1.5 D3D4 linker. And the aim of that was to do some voltage clamp fluorometry, in short VCF. So for those who are not familiar with this technique, it's an electrophysiological technique that combines a two-electrode voltage clamp setup associated with an optical system. And this allows to follow ionic change and the current, which give information about the functional state of the channel, and simultaneously to follow variations of the fluorescence signals that can be associated with molecular motion. So this allows to do structure-function relationship simultaneously for a channel that is expressed in a living cell, so in the membrane of an oocyte. But for this, NaV1.5 need to be functionalized with a fluorescent probe. And we have again used tandem protein transplicing to express fluorescence NaV1.5. And this time, the peptide X has been synthesized with a rhodamine that is covalently linked to lysine that is just upstream of the IFM particle. This represents, to our mind, a technological advance because with this technique, it's possible to label the intracellular domain of NaV1.5 with bright and photostable fluorescent probe. And so far in the literature, there is only few studies on voltage clamp fluorometry on intracellular domain of ion channel. And they have been mainly done with the method of stop codon suppression to incorporate a natural fluorescent amino acid. So this method of tandem protein transplicing, it really complements this approach. And thus, we have reconstituted the fluorescence nav 1.5 with TPTS, and we have used voltage confluorometry to follow current and fluorescence simultaneously. And here there is a representative recording. Here you can see that at a pulse at minus 80 millivolts, there is no current that is evoked and also no fluorescence change. At minus 55 millivolts, you can see that there is also no current, but there is a fluorescence signal that starts to emerge. This is a dequenching of the fluorescence. And at minus 45, there is a current and a fluorescence, which is the same at minus 35. Current and fluorescence can be recorded. And what is interesting here is that this fluorescence, this dequenching, it remains stable during the entire depolarization pulse, while the current is really fastly inactivating. So then with this recording, we have quantified the kinetic of activation, so this part, the kinetic of inactivation, and the onset and offset or decay of the fluorescence. And we have done that by mono-exponential fitting of recordings at minus 30 millivolts. And it has led us to see that the fluorescence onset, so here, was slower than channel activation, as you can see here also. Also that the fluorescence decay was slower than current inactivation, and that fast inactivation, so this kinetics here, and the fluorescent onset were having a comparable timescales. So it seems that the fluorescent signal report a motion that is kinetically related to fast inactivation. Then we followed the voltage dependence properties of fluorescence nav 1.5. So first we have done some activation protocols, and we have followed current and fluorescence and drew the curve for current in black and fluorescence in green. And when we compare the curve, you can see that the fluorescence is more sensitive to lower voltages than current. On the contrary, the steady-state inactivation curves, they are almost superimposed. So it led us to hypothesize that the fluorescence signals, it track a molecular motion that is not related to activations, but that is related to steady-state inactivation. Then we decided to track the impact of lidocaine on these fluorescence signals. And for that, I have been helped by Michelle Nielsen, who have done some VCF recording, and she's a postdoc in the lab. So lidocaine is an antiarrhythmic drug, which is used in the clinic, and is blocking NaV 1.5 pore by binding predominantly to the open and inactivated states. So we have used two different concentrations of lidocaine in a 3 Hz frequency protocol. And there we have seen that there is quite big reductions of the current with 500 micromolar of lidocaine, and there is not so much reductions of the fluorescent signal simultaneously. And this revealed that the lidocaine block is differentially affecting the current, so blocking the pore, and versus the D3D4 linker motions. So it seems that it does not interfere with D3D4 linker motion. But then we quantified the kinetics of the onset and offset of the fluorescent signals. And this revealed that the kinetics of the onset of the fluorescence was faster and the kinetic of the offset was slower upon 5 micromolar perfusions of lidocaine. And thus, we have directly probed that lidocaine impacts the kinetic of the D3D4 linker motions, even if it bind to the pore and not to this linker. Finally, we were interested to assess the impact of FGF protein on this D3D4 linker motion. So we co-expressed the fluorescence NaV 1.5 again, but this time with co-expressions with IFGF12A. And we looked at the impact on the kinetic properties. And there we saw that the onset and the offset of the kinetic signals, of the kinetic of the fluorescent signals, were both slower. So this revealed that the binding of FGF12A to the C-terminal domain is able to impact the kinetic of the D3D4 linker motions. To conclude on this VCF part, we have been able to follow a fluorescent dequenching for our constructs, which suggests that the D3D4 linker moves to a more hydrophobic environment. And this is coherent with the fact that the IFM is going to an hydrophobic pocket. Fluorescence signals that we follow report a slower motions that is kinetically not correlated with activations and that seems to be kinetically correlated with fast inactivation. And this molecular motion also seems to be correlated with steady state inactivations mechanism. Finally, we have been able to follow the impact of pore blocking drugs and CTD binding proteins and to highlight that they affect the kinetic of the D3D4 linker motion. And as a general conclusion, we are convinced that tandem protein transplicing is a powerful technology to perform intracellular domain study of ion channels, and that is something that is applicable to a lot of different other ion channels. And I want to say that the VCF label insertions by tandem protein transplicing and VCF in the human NAV 1.5 was quite challenging, so I've put this point in technical aspect also. As a limitation, we use an oocyte expression system. So this is quite not close to a cardiomyocyte lipid membrane. And also, this is an overexpressed system. And in the context of the phosphorylation project, we have an homogeneous system, which is the strength of the tandem protein transplicing because we can study a specific phosphosite in a non-ambiguous way, but in a physiological context, it's probably not 100% phosphorylated at one site. And on the second project, I have to say that the fluorescent label that we have chosen has an impact on the function on NAV 1.5. So this is a bit of a trade-off because we decided to use a fluorescent probe that is quite bright and photostable, and it's relatively big. So it can have a small impact on the function of NAV 1.5. So it's important to choose wisely also the fluorescent label. And as a main conclusion of this project, we have proved that this Y1495 phosphorylation site calibrates the FGF effects on NAV 1.5. And this family is really important for NAV 1.5 modulation. So we think that this is something that is interesting. And we have been able to perform the direct investigations of the intracellular conformational changes of NAV 1.5 in response to voltage, and also to see how these conformational changes they are affected by proteins and drugs. And to finish, I would like to acknowledge the people that have participated to this project, namely Michelle Nilsson, who have done some VCF recordings, Jacopo Galeano and Vita Sereka Bejder, who have performed the peptide X synthesis, and also Stefan Pless, my supervisor, and everyone in Stefan Pless lab. And also our molecular dynamic collaborators, Ben Corey at the University of Canberra and Reato Jean. And I would like to thank you for your attention.
Hi, Laurie. Thank you very much. Beautiful data and really cool techniques. We have a couple of questions in the Q&A. If anyone else would like to add more, they're welcome. Some more coming in now. I'll start a question from Esteban Suarez Delgado, who says, thank you so much for your neat presentation. Did you have the opportunity to study also the kinetics of inactivation and the activation curve? And I think that question was referring to the FGF study.
Yeah, so we have done also the activation for all the conditions that I have shown you, and we have not seen any significant differences on activation curves. Yeah.
And the kinetics of inactivation as well.
Yeah, we have not done the analysis yet on the kinetic of inactivations, but we have not seen obvious differences also.
We have a question from Paul Wagner or Wagner. Can you speculate why the FGF12A binding slows down the C-terminal domain motion? Having less late current with slower motions makes this very surprising.
Can you repeat the question, please? The
Yes.
FGF12, yeah.
Yeah, FGF12A binding slows down the C-terminal domain motion. Having less late currents with slower motions makes this very surprising.
I'm not sure that it slows the C-terminal domain motions, but what we see from the molecular dynamic simulations is that it increases the relative distances that can adopt the C-terminal domain versus the D3D4 linker. But in terms of kinetic, I don't know the kinetic of this interaction. Yeah.
Okay, maybe Paul can follow up if he would
Yeah.
like. A question from Damien Bell who says, "Thank you, Dr. Peverini and colleagues in the PLES lab. Lovely work. Your voltage clamp fluorimetry studies with lidocaines show poor block, but not
Mhm.
D3, D4, VSD motions. Have you
Mhm.
tried the corollary, i.e., voltage sensor domain locking toxins that would show that voltage sensor domain locking effect is correlated with voltage clamp fluorimetry effects?"
It could be really, really cool to try that. We really would like to try this kind of experiment to try to block some of the VSD. We have started to do some experiments with the protoxin 2 that are supposed to affect, I guess, the VSD2 and to have an effect on activation. But it could be really cool also to have a toxin that can block VSD4. I don't know if it was the question exactly, but yeah.
Yeah, I think that gets there. Yeah, yeah, great.
Mm.
A second question from Esteban. Usually, it is thought that the dequenching of fluorescent probes bound to the voltage sensing domain S3, S4 is produced by the upward movement and the
Mhm.
distancing from the quencher on the membrane.
Yeah.
Furthermore, it is thought that the opposite happens on the fluorescent dyes bound on the internal loops. However, in your interesting results, the internal loop dye present dequenching. Do you have any comment about where could be the quencher on the molecule?
So I think in this case, it's more like the D3D4 linker. It's so the dye that we placed, it's really really close to the IFM particle. So the IFM particle will go to a hydrophobic pocket. So this will change the local environment, and the more hydrophobicity will lead to a higher fluorescence, so a dequenching of the fluorescence, to my mind. But we don't have, of course, direct evidences to look at that. What could be cool to do would be to introduce some tryptophan in order to do some tryptophan induced quenching to more discriminate the exact part of the channels that are involved in these motions, but it would affect too much, I think, the function of NAV 1.5 in this part of the channel.
Okay, thanks. Esteban says, "Thank you for your answer."
Mm.
Susie, do you have a question?
Yeah, actually, I wanted to follow up on Esteban's question as well in terms of the VCF. So your VCF looks beautiful. I really love this technique of adding the rhodamine in such a way that you get the benefits of the rhodamine with your limitations, which very well stated.
Mhm.
But I'm curious about your interpretation of the increase in the fluorescence or dequenching, as you're calling it, being
Mhm.
hydrophobic. Right. So I'm just wondering if you could elaborate a little bit on your logic for it being, I mean, we know that the environment changes, but I'm just wondering why you thought it was specifically moving towards a hydrophobic.
Yeah, it's as I've said, it's because I expect because it's really close to the IFM particle, and during an activation, it's going to this hydrophobic pocket. So there is a lot of hydrophobic residues around. So I suspect that it's more in the fluorophore is more how can I say oriented toward the more hydrophilic environment, and then when there is an activation, it moves towards the close to the hydrophobic pocket of the IFM particle.
Are there studies showing that rhodamine specifically increases in fluorescence in hydrophobic environments?
Yeah, yeah. The fluorescence of dye usually it's increased in a hydrophobic environment. Yeah.
Okay,
Mm.
great. Thank you so much.
Laurie, you mentioned it in your technical aspects here. I wondered if you could elaborate on how difficult it is to find these insert sites. Do you have to screen a wide number of different sites? Is there any logic to where you can target with this approach?
So you mean to design the intended construct? Yeah.
Right.
Yeah. Yeah. So we use, in fact, in the lab, we use already optimized couple of split inteins. And these, they need a specific sequence to be really efficient in their splicing reactions. So you might need, when you want to incorporate this system into your protein of interest, you might need to mutate some residue. So you need to do the control to see if the residue that you mutate, they already change the function. But yeah, usually you need a cysteine or a serine or nucleophilic residue in order to implement this technology. So if you don't have it in your sequence, either you screen your sequence and you select the part where there is this type of residue, or you can just mutate these residues in order to have an efficient splicing reaction. And then what is important also is to find a couple of, how can I say, asymmetric inteins. So I can maybe show a slide on that. So yeah, because these split inteins, they have two parts. And what is important to have a synthetic peptide that is possible to synthesize, you need to have some quite small C part of the first split intein and N part of the second split intein. Yeah. So this is something to think before doing the experiments, but then it's possible to design it. And there is several examples now on several ion channels that are amenable to use this technology, and the splicing is quite efficient. Of course, less efficient than wild-type proteins, but it worked.
Yeah.
I don't know if it
Are
was
there
exactly
limits
the
to
question.
That's right. Yeah, that got it. Are there limits to the size of the peptide that you can insert? Do you get a trade-off for efficiency if you try and insert longer peptides?
I don't think that it's on the efficiency of the splicing, but it's more that at one point the synthesis. So the synthesis of the peptide is done by solid phase synthesis. So at one point the peptide, when it's too long, you will not have a good purity, and it will be quite painful for the person who needs to synthesize the peptide. So it needs to stay in the range of like less than 100 amino acid, I guess. So you already have almost 40 amino acid for the split intein that I have shown you before. It's more than 40 residues. So then the part of the channel that you can put is there. It's, yeah, around 20, 30, 40 amino acid. Yeah.
Really, really cool technique and lovely data. I guess if we don't have any more questions, we can say thank you very much for presenting. So well done,
Thank you
Laurie.
so
That was
much.
really, really brilliant. And I'd just like to say to everyone, you're welcome to join us again on the 2nd of June when we have a presentation from Wojciech Kopek, which will be about molecular dynamic simulations of membrane proteins. So thank you everyone for joining. Thank you, Laurie.
Thanks a lot.
Thanks a lot.
Thank you.
Well done.